An algal bloom or marine bloom or water bloom is a rapid increase in the population of algae in an aquatic system. Algal blooms may occur in freshwater as well as marine environments. Typically only one or a few phytoplankton species are involved, and some blooms may be recognized by discoloration of the water resulting from the high density of pigmented cells. Although there is no officially recognized threshold level, algae can be considered to be blooming at concentrations of hundreds to thousands of cells per milliliter, depending on the causative species. Algal bloom concentrations may reach millions of cells per milliliter. Colors observed are green, yellowish-brown, or red. Bright green blooms may also occur. These are a result of blue-green algae, which are actually bacteria, cyanobacteria. Some algal blooms are the result of an excess of nutrients, particularly phosphorus and nitrogen, into waters, and higher concentrations of these nutrients in water cause increased growth of algae and green plants. As more algae and plants grow, others die. This dead organic matter becomes food for bacteria that decompose it. With more food available, the bacteria increase in number and use up the dissolved oxygen in the water. When the dissolved oxygen content decreases, many fish and aquatic insects cannot survive. This results in a dead area. Algal blooms may also be of concern as some species of algae produce neurotoxins. At the high cell concentrations reached during some blooms, these toxins may have severe biological impacts on wildlife. Algal blooms composed of phytoplanktons known to naturally produce biotoxins are often called harmful algal blooms, or HABs. Domoic acid is produced by algae and accumulates in shellfish, sardines, and anchovies, which are then eaten by sea lions, otters, cetaceans, and humans, among others. Exposure to the biotoxin affects the brain, causing them to become lethargic, disoriented, and have seizures that sometimes result in death. It is becoming increasingly clear that marine mammals are sentinels of potentially dangerous environmental changes in the ocean environment. Sick and stranded marine mammals warn us of changing ocean conditions in El Niño years, and poisoned marine mammals have protected human health by demonstrating the need for the screening of shellfish and other seafood for fatal biotoxins. The Center's Veterinary Science Program, under the direction of Dr. Francis Galland, senior scientist at the Marine Mammal Center, lays the foundation for a deeper understanding of human behaviors that may affect marine mammal survival for the long term. Research outcomes lead to a clear understanding of overall risk factors contributing to disease in marine mammals, and provide vital information to resource managers in the San Francisco Bay Area that can be used for informed mitigation of human activities impacting marine mammals. Additionally, our research program directly informs our education initiatives, which provide resources for the public to deepen its understanding of threats to the health of California's coast and how to participate in protecting this environment. Investigation into domoic acid toxicity. Often referred to as red tide, domoic acid, DA, is produced by a harmful algal bloom. DA can have devastating effects on marine mammals and humans that consume toxic prey species. Severe cases of seizures and other central nervous system problems, as well as hippocampal degeneration and amnesiac shellfish poisoning in humans, have been documented as caused by DA. Further study may reveal potential risk factors that cause DA poisoning outbreaks amongst California sea lions, and help point the way toward preventative measures. Domoic acid is a neurotoxin produced by phytoplankton, specifically a microscopic diatom, Stenotyschia australis, pictured at the top of the page, in the ocean. This toxin causes seizures in higher vertebrates as it concentrates up the food chain by targeting the brain, specifically the hippocampus. California sea lions and other marine mammals become affected when they eat prey, like anchovies, that have been feeding during toxin-producing algal blooms. The effect of DA on sea lions depends on the amount they eat. Diagnosis depends on detection of the poison in serum, urine, or feces of affected animals, coupled with detection of P. australis in the environment and prey of affected sea lions. As DA is water-soluble and rapidly excreted in urine following ingestion, urine is the most useful fluid for diagnostic purposes. Because of the variable toxicity of algal blooms and the unpredictable timing of DA outbreaks, diagnoses are typically difficult to establish definitively, and many other factors must be ruled out in the process. The Marine Mammal Center was the first group to definitively diagnose DA poisoning in marine mammals because of a large outbreak in California sea lions in 1998. In September 2004, the center received a grant from the Oceans and Human Health Initiative to study the long-term effects of domoic acid in sea lions. This project studied the impact of DA on health, survival, and reproduction. Part of this project focused